السلام عليكم. أنا الطالب ركان حداد من كلية الطب في الجامعة الأردنية. Today we're going to be explaining the final lecture of Neoplasia. Epstein-Barr virus. نكمل هلا على اللي كنا نحكي عن microbial أو microorganisms that cause cancer. هلا نكمل. Epstein-Barr virus is part of the herpes virus. The first one to have ever been linked to a human tumor. فبيجي بيسألك what is the first one to have ever been linked to a human tumor؟ تحكي له Epstein-Barr virus. بيعمل إشي اسمه Burkitt's lymphoma. It's actually really aggressive. It's considered an endemic in Africa, okay? In Africa it's an endemic. لكن anywhere else it's considered sporadic يعني بصير هيك يعني it can happen. In endemic areas, the tumor cells in virtually all affected patients, they have the Epstein-Barr virus genome, okay? In an endemic area, يعني in a place that we know that the virus is present in a large amount. Any Burkitt's lymphoma will tend to have EB virus genome. Okay, any patient with Burkitt's lymphoma is going to actually have EBV genome. Okay, mostly. لكن if we're talking about مثلا in the United States and we want to test the people with Burkitt's lymphoma, the Epstein-Barr virus, the genome of it might not be present in all of them. Okay, maybe in just 2%. Who knows? فبس بيحكي لك كون انه في مناطق معينه لانه الفيروس منتشر هناك اكثر because the virus exists more. يعني the lymphoma, the Burkitt's lymphoma is going to be associated to it more. Epstein-Barr virus has been implicated in the pathogenesis of other tumors or other cancers. إذا واحد هو Burkitt's lymphoma حكيناه. Lymphomas in immunosuppressed individuals مثلا people with HIV or people who took organ transplantation. أي حد مع HIV معرض يصير له AIDS اللي هي advanced immunodeficiency. People with organ transplantations لما نعطي حدا أو لما نعمل organ transplantation بشخص, he basically took something out of another person's body. This is considered a weird foreign body that. فزي ما كنا نحكي, people who would get an organ transplantation, we have to supply them with steroids. ليش? Because the body will consider this as a foreign body. Okay, it's a foreign body, so our body, our immune system is going to attack it. Okay, ما بدنا هذا الحكي يصير. So we give steroids. When we give steroids, we're putting this patient in an immunocompromised state. طيب. برضو Hodgkin's lymphoma, nasopharyngeal, T-cell, gastric, and sarcomas, and mainly in immunosuppressed people. The Epstein-Barr virus tends to attack B cells. كيف? It attaches to CD21 complement receptor. The infection causes polyclonal B cell proliferation. شو يعني? B cells, they can form monoclonal or polyclonal. Monoclonal means that they will give one type of antibody. Okay, one type of antibody. Polyclonal, they will produce multiple antibodies. That's the whole point. Okay, multiple antibodies. And it will end up generating immortal B lymphoblastoid cell lines. Hello, there is a specific gene, it's an oncogene actually in the EBV, in the genome يعني. It's called latent membrane protein 1, LMP1. It's actually the oncogene here. طبعا the gene product اللي هو LMP1, it contributes to oncogenesis by stimulating a normal B cell proliferation pathway. شو اللي انت قاعد بتخبصه هون؟ Basically, the EBV gene هون, it caused the polyclonal proliferation of B cells by activating normal B cell proliferation. طيب, LMP1 بيشتغل or خلينا نحكي, it stimulates the signaling by NF nuclear factor kappa B and the JAK-STAT pathways. Okay? In both of them, they promote the B cell proliferation and survival. هذا بحكي لك هي إنه what's happening here is a normal proliferation pathway. يعني we're studying a normal pathway. شو هي? Nuclear factor kappa B activation or JAK-STAT pathways. وين المصيبة بتيجي؟ لما يصير عندك prevention of apoptosis. Okay? Through mean? Through the BCL2. Okay? Through the BCL2. طيب, we already know what that is. فعشان نختصر وقت, you can go review it from the other lectures. Epstein-Barr virus gene EBNA2, it activates cyclin D. شو بيعمل cyclin D? It activates the cell cycle. بس هي. VIL-10 prevents macrophages and monocytes from activating T cells and killing the infected cells. إذا نفس ال B cells اللي صار لهم infection, the B cells that got infected, they won't die. Okay, they're gonna become immortal. حكيناها. They will نفس الفيروس generates immortal B cells. Okay, these B cells are gonna proliferate. كيف? The normal pathway. شو هي normal pathway? Nuclear factor kappa B أو JAK-STAT pathway. وين المصيبة؟ inhibition of apoptosis by BCL2. حلو. مين هذا كله بيعمله؟ LMP1. إجانا جين تاني, another gene got introduced, EBNA2, activates cyclin D لحتى تكمل عندي cell cycle. VIL10, it prevents the macrophages and monocytes from activating these T cells. هذا اللي هو بيعمل الامورتاليتي لهم. Okay, this gives me the immortality. In regions of the world where Burkitt's lymphoma is endemic, concomitant infections. Okay, مثلا malaria is going to impair the immune competence. ايش اللي بيصير؟ people that have Burkitt's lymphoma as an endemic, يعني بسبب انهم تواجدهم بمنطقة, because they're located in a specific place, let's say Africa. Okay, people in Africa are also, يعني they can get malaria. Malaria is going to impair the immune competence. What will that cause? It will even cause more damage. ليش? Because the T cells are going to even get weaker هلا. Okay, so the B cells are going to proliferate كأنه فيش شيء عم بصير, and this is obviously really harmful. لكن eventually بيحكي لك cytotoxic T cells will be able to eliminate most of the EBV infected B cells, لكن a small number can survive. Okay, concomitant compromise of immune competence allows sustained proliferation. ليه? Because they're basically hiding the infected cells. ليه؟ لأنه في infection تاني بدنا نتعامل معه. Okay, so because there are two infections, we want to deal with one first then the other. Let's understand it in that way. يعني خلينا نحكي. Okay, طيب. And eventually the development of a lymphoma. Okay, more mutations are going to occur. One of them is the T814. If you don't know what that is, you can also go back to the previous lectures. بكل بساطة يعني it involves different chromosomes, where a part here is يعني بيجي يعني part of the chromosome goes to another part of another chromosome and so on. يعني it's not that difficult. Anyways, holiday is it activates the MYC gene. Okay, it leads to the activation of the MYC gene. هذا regulator. It's also considered a tumor suppressor أو خلينا نحكي a growth regulating gene. Okay, يعني if you wanna if the cell wants to proliferate, if it wants to grow, the MYC gene needs to be activated. Okay, طيب. But what they're telling me here is this mutation causes activation of that, any more proliferation, survival, and so on. In non-endemic areas, 80% are unrelated to EBV, as I already explained before, but virtually all endemic and sporadic tumors, they have this translocation, okay? Or other translocations that dysregulate the MYC gene. Then we must understand the MYC gene is of high importance due to a translocation mutation between chromosome 8 and 14, maybe another chromosome, and here they're telling us, or other translocations that ممكن يصير, تمام? So يعني we must understand that 80% of the tumors, Burkitt's lymphoma, they have nothing to do with Epstein-Barr virus. يعني Burkitt's lymphoma in non-endemic areas, okay? لكن the mutation that happens, the translocation, it's always present in this sort of lymphoma, okay? Sporadic Burkitt's lymphoma are triggered by mechanisms other than the EBV infection, مو شرط, but they appear to develop through similar oncogenic pathways. Then eventually, the development of the Burkitt's lymphoma is similar whether you got it from whether someone got it from Epstein-Barr virus or sporadically. Okay, تمام. So we must understand that the pathways triggered are similar. تمام. The genes, the translocations, the mutations, everything is kind of similar. إلا إيش؟ إلا the first خلينا نحكي the first mechanisms that occur. Okay. Nasopharyngeal carcinoma is also associated with it. This tumor is endemic in southern China and in some parts of Africa. Nasopharyngeal carcinoma, اعرف انها endemic وين? Southern China and some parts of Africa. بيجيك واحد من China, southern China. He has nasopharyngeal carcinoma. You have to suspect Epstein-Barr virus. You have to. In contrast to Burkitt's lymphoma, 100% of nasopharyngeal carcinomas obtained from all parts of the world contain the Epstein-Barr virus. Okay? برضه بيحكي لك إنه مو شرط just in southern China مثلا. لا. From anywhere in the world, people with nasopharyngeal carcinoma, they actually have Epstein-Barr virus. Okay, so this tells you how dangerous this virus is. بس Burkitt's lymphoma 80% من اللي in non-endemic areas they don't have that virus. Okay, so yeah, these are really important statistics. You have to memorize them. تمام. That's it. It's really important to know about in southern China and some parts of Africa, nasopharyngeal carcinoma mainly due to the Epstein-Barr virus infection. Okay. ومنتشر أكتر يعني خلينا نحكي بهاي المناطق southern China and some parts of Africa. Okay. But طبعا بالstatistics هاي بتحكي لك إنه if you get someone he has nasopharyngeal carcinoma, he most likely has Epstein-Barr virus. Okay. The integration site of the viral genome is identical, clonal, in all of the tumor cells within individual tumors. Okay, we already talked about that in I think the previous lecture when we talk about a specific tumor, هذا التومر المعين, it's always going to integrate its genome. طبعا if it's from a virus يعني, it's always going to integrate its genome at a specific part. طب tumor تاني at another part. Okay, so this is the whole idea يعني إنه it's identical in all tumor cells within individual tumors. Unlike Burkitt's lymphoma, LMP1 is expressed in nasopharyngeal carcinomas cells and in B cells that activate the nuclear factor kappa B pathway. Nuclear factor kappa B upregulates the expression of factors such as the VEGF, a growth factor, and matrix metalloproteases. They may contribute to oncogenesis. بكل بساطة, the pathway is activated here by the nuclear factor kappa B will cause more growth factors to arrive and will cause matrix metalloproteases. Okay, دول درسناهم بالريبير وبرضه حكينا عنهم بمحاضرة. نسيت صراحة أي وحدة. Matrix metalloproteases, and these will contribute to oncogenesis even more. They will help in metastasis and so on. ومعلومة نحكيها مرة تانية إنه the LMP1 هذا it's expressed in nasopharyngeal carcinoma cells, okay, and in B cells that activates the nuclear factor kappa B pathway. Then this gene is not expressed in Burkitt's lymphoma. Okay, بدنا نعرف هذا الشيء. هلا حكينا عنه ال LMP1. Okay, بيعمل هذا الحكي كله. It's not expressed in Burkitt's lymphoma. تمام. ثالث فيروس هو الهepatitis B وhepatitis C viruses اثنيناتهم. Okay, 70 to 85% of hepatocellular carcinoma, they're actually due to the infection of hepatitis B and hepatitis C. Okay, statistics مهمين هذول. The oncogenic effects are multifactorial, but the dominant effect seems to be immunologically mediated chronic inflammation. فصار في injury, okay, and as a result, proliferation occurred in the production of ROS, and these ROS damage the DNA. حلو. إذا شو اللي صار؟ بكل بساطة, the damage that the virus does to the liver is going to cause hepatitis, اللي هو the inflammation. Okay. لما يصير chronic inflammation, هون dangerous cases, chronic inflammations, this can cause injury. حلو. الانجري we want to repair this injury. هلا أصلا while the injury is present, there are lots of reactive oxygen species present. Okay, ليه? Because we want to remove the virus. Okay, we want to remove whatever is causing that. Okay, anyways, we will stimulate hepatocellular proliferation and production of ROS is going to occur. This ROS damages the DNA. شو يعني damages the DNA؟ بيعمل mutations. طب شو يعني mutation؟ يعني cancer. وبهاي الحالة hepatocellular cancer. The HBX protein of HBV and the HCV core protein can activate a variety of signal transduction pathways. They may also contribute to carcinogenesis. اللي هم زي antigens of these. Okay? شو بيعملوا؟ These antigens, they cause a signal transduction pathway that has to do with carcinogenesis. As with any cause of hepatocellular injury, chronic infection leads to the compensatory proliferation of hepatocytes. أي hepatocellular injury يكون chronic viral infection leads to the compensatory proliferation. I want to compensate for the part of the liver that's dying or that's damaged by proliferation, okay, of immune hepatocytes. This regenerative process is aided and abetted by a plethora of growth factors, cytokines, chemokines, and other substances. مين اللي بيعمل production؟ immune cells. طبعا هذا كل الحكي عارفينه يصير cell survival, remodeling, angiogenesis, and producing a new part of the liver. Okay, بس فكل هذا أخذناه زمان. So don't يعني just read it يعني. The activated immune cells, they can produce other mediators اللي هم ال ROS. هذول they're genotoxic, mutagenic, carcinogenic, شو ما بدك سميهم. A key molecular step seems to be the activation of the nuclear factor kappa B pathway in hepatocytes caused by mediators derived from the activated immune cells. إذن نفس immune cells, they're going to start secreting mediators, and from these mediators they're going to activate the nuclear factor kappa B pathway. Okay, شو بيعملوا؟ They're going to activate the nuclear factor kappa B pathway. Okay. And when you activate the nuclear factor kappa B pathway, شو اللي عم تعمل؟ You're blocking apoptosis. And this is going to cause genotoxic stress and the accumulation of mutations. ليه؟ ال ROS لسه شغالة. The ROS are still working. What are they doing? They're producing mutations. They're destroying the DNA. Okay? They're destroying the DNA. Okay, they're destroying the DNA. So what? We're going to get rid of these bad cells. لا. Because the nuclear factor kappa B pathway was activated, the cells that are getting damaged are staying alive. Okay? نكمل. The hepatitis B virus genome contains a gene known as HBX. HBX can directly or indirectly activate a variety of transcription factors in several signal transduction pathways that interfere with the P53 function. إذا HBX هذا جين شو بيعمل HBX protein حكينا عنه قبل شوي. Okay. نفس ال HBX, it has the ability to directly or indirectly, doesn't really matter, to activate transcription factors حلو and several signal transduction pathways. What will they do? They will interfere with the P53 function, which is a tumor suppressor gene. Okay. إذا the hepatitis B virus has HBX هذا جين بيعمل protein called HBX. This protein is going to activate specific transcription factors and signal transduction pathways in which it will interfere with the normal P53 tumor suppressor gene. Okay. طبعا برضو viral integration can occur. بيمسك حاله ال DNA virus وبيدخل على normal genome. Can cause secondary rearrangements of chromosomes. بكل بساطة it ruined the original organization, the original nucleotide sequence. It caused even worse mutations when it integrated. Okay, may cause deletions, may cause lots of other mutations. Okay, and that may harbor unknown tumor suppressor genes. These deletions can maybe accidentally delete a wanted gene مثلا. Okay, بالغلط انت رحت شطبت tumor suppressor gene. This is really bad يعني. Helicobacter pylori, this is a bacteria. It causes gastric adenocarcinoma and the Malt lymphoma. The mechanism of the Helicobacter pylori-induced gastric cancers is multifactorial, including immunologically mediated chronic inflammation. زي مين بتذكركم؟ Obviously hepatitis. Okay? إذن for it to cause gastric cancers, لازم يصير في inflammation. Okay, inflammation, chronic inflammation. With this inflammation, what will happen? Proliferation. ومعنى proliferation والinflammation أهم شيء طبعا من وراء الانفلاميشن والrepair في عندك ROS. Okay, these ROS will damage the DNA. Helicobacter pylori pathogenicity genes such as the CAG-A may also contribute by stimulating growth factor pathway. Then also we have other genes. One of them is the CAG-A. This gene is going to stimulate the growth factor pathway. Okay, the stimulation is going to cause increased epithelial cell proliferation on a background of chronic inflammation. انت بكون عندك inflammation وعلى الانفلاميشن هذا you're having proliferative cells, lots of proliferative cells. Okay. طيب. The sequence of histopathologic changes consists of initial development of chronic inflammation and gastritis, followed by gastric atrophy, intestinal metaplasia of the lining cells, dysplasia, and cancer. And بقول لك إن you have a specific sequence that occurs. Okay, there's a specific sequence that occurs. Okay, and it's really simple. يعني you have specific changes. أنت بلشت بinflammation. This inflammation caused gastritis. Okay, suddenly gastric atrophy took place. Then intestinal metaplasia it became dysplasia, and finally caused cancer. Okay. This sequence takes decades to complete and occurs in only 3% of the people that are infected with Helicobacter pylori. Okay? So يعني بدها وقت كتير عشرات السنين and it only infects 3% of the people that are infected with H. pylori. تمام? Strains associated with the gastric adenocarcinoma, they have to contain pathogenicity island. شو يعني pathogenicity island? Let's consider it a part of the Helicobacter pylori DNA. Okay, it's like the circular DNA of the bacteria. Let's consider the pathogenicity island is basically an island of genes, an island of carcinogenic genes. Okay, that contains cytotoxin associated A gene اللي هو الكاج A اللي حكينا عنه. تمام؟ Okay, اللي هو اللي بيعمل growth factors and so on. CAG A is injected into the gastric epithelial cells. This will cause إيش؟ Proliferation. Okay, من وراء the growth factors. H. pylori is also associated with increased risk of developing gastric lymphomas. The gastric lymphomas are of B cell and referred to as malt lymphoma. Okay, إذا حكينا إما gastric adenocarcinoma أو malt lymphoma. المالت lymphoma هي عبارة عن شو؟ المالت lymphoma هي عبارة عن gastric lymphoma. Okay, because they are of the B cells. Their molecular pathogenesis is incompletely understood, but seems to involve strain-specific H. pylori. Okay? يعني بكل بساطة strain specific depends on the type of the Helicobacter pylori that did the attack or did the whatever. Okay? طيب. H. pylori infection leads to the activation of the Helicobacter pylori reactive T cells, which in turn cause polyclonal B cells and the malt lymphoma. Okay? So really simple, there's nothing really difficult here. Basically, there are specific T cells called H. pylori reactive T cells. These reactive T cells are gonna cause polyclonal B cell and malt lymphoma. Diagnosis of cancer in the lab, you can use morphologic methods, immunocytochemistry, molecular diagnosis, flow cytometry, tumor markers, and yeah, we'll go back to the rest of it. Anyways, morphologic methods, انك انت بدك تشوف شكل, you want to see something, okay? You want to see the shape of the tissue, and then you can decide. You can use hematoxin the Yosin stain, HND. You can do an excision or a biopsy, okay? Fine needle aspiration, cytologic smears, okay? And frozen sections. So if you want to do morphologic methods, you can use your HND stain, and you can stain you a specific biopsy, whatever you want, يعني, okay? مكتوبين هون really easy. Immunocytochemistry, you can use cytokeratin, prostate specific antigen, PSA. بيجي بيسألك what can I do to be able to diagnose cancer in a prostate carcinoma in a lab? You can use the PSA, prostate specific antigen. So what about breast cancers? You can use estrogen receptors, okay? HER2. طيب. Molecular diagnosis, okay? Estrogen receptor, okay. Anyways, diagnosis of cancer, CML, PCV. Prognosis and behavior, the HER2 and the NMYC. Detection of minimal residual disease. Okay, by the way, when they talked about immunocytochemistry, we want to target specific things like the estrogen receptor in breast cancers, okay? We're going to target the receptor. In molecular diagnosis, I'm noticing the activity of the receptor. فنعرف نفرق بيناتهم, okay? Detection of minimal residual disease, diagnosis of hereditary predisposition to cancer. You can look at specific genes, look for mutations. These are molecular خلينا نحكي methods. Flow cytometry, it's used in the classification of leukemias and lymphomas. Fluorescent antibodies against cell surface molecules and differentiation antigens are used to obtain the phenotype of the malignant cells. Okay? Tumor markers, حكينا في أشياء بتورجيك إنه في tumor مثلا. Okay? The PSA, prostatic carcinoma can be suspected when elevated levels of PSA, we can find that in the blood. Okay? So by the increased amount of PSA, we can tell that there is مثلا prostatic cancer. Although PSA levels are often elevated in cancer, PSA levels also may be elevated in benign prostatic hyperplasia. Okay, this is sort of hyperplasia. We took it. It's benign. So the thing here is that the PSA test مش هتعطيك نتيجة كاملة. It has low specificity. Okay, so first of all, the PSA test of low sensitivity and low specificity, because the elevated PSA can either indicate that a person has prostatic carcinoma or benign prostatic hyperplasia. Again, let's talk about why is PSA mentioned in the tumor markers and the immunocytochemistry. In immunocytochemistry, we're targeting the antigen itself. Okay, I wanna be able to visualize it. I wanna see. The tumor markers هم عبارة عن tests we can do to measure the amounts of something in the blood or whatever to know if they're elevated or not, and so on. The second test, carcinoembryonic antigen, CEA. Carcinomas of the colon, pancreas, stomach, and breast. Okay, we're looking for the C-alpha fetoprotein. This is produced by hepatocellular carcinomas, yolk sac remnants in the gonads, teratocarcinomas, embryonal cell carcinomas, neural tube defects of the fetus. And the CEA and the alpha fetoprotein assays lack both specificity and sensitivity. Grading and staging of cancer methods to quantify the probable clinical aggressiveness of a given neoplasia and its apparent extent and spread in the individual patients are determinants of prognosis and treatment protocols. Then we must understand that CEA and alpha fetoprotein, these are really weak. They lack specificity and sensitivity to be able to grade and stage cancer. زي ال PSA لسه أضعف حتى PSA low sensitivity, low specificity because we're comparing two things. هون مليان شغلات you might not know what's going on. Then again, let's repeat it real quick. Laboratory diagnosis, we can look for shapes. نشوف أشكال. كيف نشوف أشكال؟ شو بتخبص؟ I want to look at a specific tissue and be able to understand what's going on in that tissue. So I can use stains, okay? I can stain biopsies, smears, whatever you want, okay? الصورة اللي عندنا هون, this is papina solu, okay? We used اللي هي cytologic smears هون, تمام? This is an example, we use like, what method did we use here? We use morphologic methods by staining, okay? Okay, عندك by using a cytologic smear immunocytochemistry هي عبارة عن شو? Basically I wanna attach something to the receptor. Okay, immunocytochemistry. I'm looking for a specific receptor or a specific protein, and I'm targeting it by an antibody or an antigen. Okay. Molecular diagnosis. حكينا we're looking for elevated levels. Okay, elevated levels of specific things. Molecular diagnosis. I'm looking for molecular effects. I'm looking for genes. I'm looking for molecules. I'm looking for whatever. Okay. Sorry, تخربطت بالتحت. Okay. Flow cytometry. That basically I'm using fluorescent antibodies against the cell surfaces, and differentiation antigens are used to obtain the phenotype of a specific malignant cell. Okay, and بجيب antibody, هذا antibody بضوي, I target it on a specific cell, and then I use differentiation antigens so I know what tissue is this from. تمام? Tumor markers, I'm looking for elevations in the blood, maybe PSA for prostatic cancer, carcinoma, or benign حسب, it has low specificity, low sensitivity. The C-alpha fetoprotein وحكينا it is أو it lacks specificity و sensitivity. هلا grading and staging of cancer. Grading هي مش لما واحد يحكي لك stage 1 stage 2 لا ما لهاش دخل. It's different. Grading has to do with the level of differentiation. Okay. when I say grade مثلا grade 1, grade 2, grade 3, grade 4, I'm talking about the level of differentiation. Staging هذا هو الواحد نتعامل فيه أكتر لما يجي واحد يحكي والله you have stage 4 cancer, stage 3 cancer, stage 2 cancer, whatever, okay? So, grading, it's also into 4 categories. 1, 2, 3, 4. It used to be 2. كانوا بس يحكوا low grade أو high grade زمان. هلا صار يحكوا grade 1, grade 2, grade 3, grade 4. اوكي مثلا well differentiated mucin secreting adenocarcinoma of the stomach, poorly differentiated pancreatic adenocarcinoma. مثلا well differentiated I guess like grade 1, poorly differentiated grade 4. staging لا staging is more complex, تعتمد على 3 أشياء. depends on 3 things. ايش هم؟ size of the primary lesion, how it spreads to the lymph nodes, وبرضه presence or absence of metastasis. Okay, size of the primary lesion, we can give it a size from T1 to T4. Okay, to T4 بين T1 و T4. تمام؟ أكيد whenever you're مثلا T4 is larger than T1 and so on. يعني بدهاش شيء كتير صراحة. طبعا this system is used in the American joint more يعني when we watch movies or series جديدة هاي. سوري الساعة 1 مش نايم، صار لي 3 أيام ورا الامتحانات فالله بيعينكم علي. Anyways, when you're watching a specific movie or a series, medical series, تسمع والله this guy has stage 3 cancer, stage 4 cancer. And staging is the خلينا نحكي most common يعني system. The Committee on Cancer Staging في عندك TNM system اللي هي T for primary tumor, N lymph node involvement, M metastasis. T يعني size. Okay, T1 أصغر شيء، بعدين T2 T3 T4. N regional lymph node involvement، إذا كانت N0 يعني no lymph nodes are involved. Okay، وفي N1 وفي N يعني لحد N3. تمام. So N3 has more more lymph node involvement than N2 and more than N1. Okay. في برضه M for metastasis, M0 indicates no metastasis, and we also can use M1 or M2 for the level of metastasis. بس يعطيكم العافية. هيك بنكون خلصنا Neoplasia 9 lectures سهلات إن شاء الله بتتسلوا فيهم. and good luck. إن شاء الله ما أكون قصرت معكم بأي شيء، موفقين وتجيبوا علامات عالية إن شاء الله. Goodbye.